Our privilege today to have Professor Chandra Verma with us for the Institute lecture. And to begin with, I would like to welcome Professor Chandra Verma, and I will request our director to hand over a bouquet. So, Professor Verma, he carried out his undergraduate studies at IIT Kanpur, followed by PhD at the University of York, UK. Following a postdoc and research fellowship, he relocated to the Bioinformatics Institute, IISER, Singapore, as a group leader. His group explores biomolecular mechanisms, small molecule peptide, protein design in an interdisciplinary team of experimentalists, clinicians and pharma companies. The work has resulted in a spin-off Simsa Labs, which has licensed their anti-infectives, and more recently in a spin-off Therapeutics for developing treatments for wet AMD, a degenerative age-related eye disease. So without wasting much time, I would like to welcome Professor Chandra Verma for his talk.
Thank you. Thank you very much for being here. Thank you, Uma, for this nice invitation. It's a beautiful campus. I have a long history with this institute, at least going back 8 years, when Dilraj Lama, who was from the biological sciences, came and joined the BII, and suddenly 8 years went by and we didn't realize. And he's gone to Karolinska now. Okay. So at this time, I always think that giving a lecture at this hour is torturous both for you and for me at the end of a long day, but anyway, we try and make it interesting. So I'll give you some insights into a kind of computational modeling and how it is used regularly by various groups in the world with various degrees of success. We've been lucky in some cases, unlucky in most cases, so I'll talk about the luck over here. And during the course of my presentation, if you wish to stop and just ask, please do. Okay, because I think at least for the initial phase, I'll take some time to describe the technology we use, and apologies to those who know about it. Might be boring, but at least for the ones who do not, might be a little bit of insight into what we do, the various pros and cons of these technologies, and where we go ahead with them. Some of it will reflect on this excellent talk I heard earlier on about the VEGF and the receptor and the pathways in cancer. I will come to that later. Okay. So, apologies for trivializing this a bit, but sometimes it's very nice to think of some kind of an analogy for the kind of work we do. So, if you were an architect assigned with the task of, let's say, designing a city, what are the questions you would have to ask, right? So you like to ask, why should I build a city? Right? And so a simple trivial example is to allow for some function in a protected environment, right? That's a very trivial reason, but nevertheless it's a good enough reason to go on with. And then within the city you'll have occupants, where will they live, what sort of compartments you have to think of while designing a city, how long will these people stay over there, how will they interact with each other, so on and so forth. And more importantly, in this process of building your city, sometimes things will go wrong, and sometimes things will go horribly wrong, which will require you to scratch your heads and try and figure out solutions to this, which is what we would call a disease. So a city is a good analogy for a cell at one extent. Okay? But now, once you are inside the cell, what do you have to worry about? So now imagine that this was a circus, that you were all artists in a circus. What do you have to do? Yeah? The biggest thing you have to do is, like one of your cultural festivals, right? You run a show for a few days, and then you have to dismantle everything, right? You pack it away somewhere, and then you reopen somewhere else. Right? So for those of you who attended the talk earlier on, metastasis is a good example. It's a sad example because things go very badly wrong, but it's a good example of what all a cell has to evolve to perform the kind of a function. Okay? And all of these require a very, very exquisite coordination in space and time. That's very, very key. Okay. So now here the artists are cellular molecules, okay, which work as parts of teams or complexes and cascades in time. And therefore it becomes very important to understand these interactions both in space and in time to get some idea of what biology is about. Okay. And so brief snapshot into the complexity one deals with came out in this paper in 2010 when people tried to figure out the various protein-protein interactions in a simple organism called the yeast. Okay, and how they began to understand how these multiple interactions in the yeast were part of large networks of these interactions. But the nice thing was they were able to at least annotate a fair amount of these interactions to various functions in the cell and to various compartments in the cell. That was a very interesting and nice finding, that a lot of the functions emerge from congregations of these proteins which are all compartmentalized, which makes sense intuitively, but of course, when you actually have validated it experimentally, it's quite clear. Okay. So this word interaction that I keep using, right? If you think about it, it has two components to it. One is an inter of the interaction, which really is about the spatial relationships about any two objects. Okay, so this is where you need to understand, for example, structure. And then there is the action bit of the interaction, right? And that immediately tells you there must be some dynamics in this, and therefore you need to really understand temporal relationships. So, for example, one of the cellular constants is proteins. We know that they interact physically with other molecules in the cell, and of course these interactions vary with time. And you may have, okay. So, of course, over the years, people have developed lots of biophysical and biochemical techniques, yeast 2 hybrids, etc., to try and figure out at least pairwise or now even more complex interactions between two species. And then, you do PhDs on trying to pursue, one of these interactions, for example. Okay? And then, of course, it always helps if you have structural information that backs up these findings, because it allows you to then do some rational investigations, such as specific mutational analysis, etc., etc., etc. Okay. And then once you have this data, you can begin to create these kinds of networks, right? Once you begin to develop networks, you develop databases, and then you can mine these networks to make various discoveries. And increasingly you see a lot of people using bioinformatics tools to mine and actually come out with very, very interesting discoveries. So, for example, there is this very interesting paper which I thought was quite nice. It was driven by an in silico mining of this one of these networks for cervical cancer. Okay. So then what do you do? So at the next level of complexity, then you have to ask this question, right? That once I have annotated all the information on the structures and the locations, at least to some extent, of all these various proteins in the cell, can I begin to then model how these proteins might interact, how these things move around with time, and how might function emerge out of it? Those are the three questions you would like to simulate or to try and understand, and then make very, very testable hypotheses out of it. That's very critical. And so therefore, of course, I am biased because I work in the structural domain, but it is clear that without this kind of structural data, our understanding of modern biology would be very, very, sad and very highly incomplete without this. I mean, not a day passes by without you see some publication of some nice relationship between a structural feature and a biological feature that's known. And then, of course, the developments in spectroscopies of various kinds, most latest being, very advanced resonance Raman spectroscopies, for example, which is Prof domain, and more computer modeling actually are beginning to provide a lot of very, very interesting insights into this. So then you may ask, so what are these computer models that one talks about? Of course, there are various levels of computational modeling at different scales. I will only touch upon the atomistic level of modeling, and there is also lots of computational biology, network modeling, etc. Okay. And we will use P53 as an example because P53 is something that I am a little familiar with. And then, if we have time, then we will try and see where may all this lead us to. Yeah? What are the hard questions that need to be answered in the future by the likes of you all. Okay. So this is very well known, right? That you have a string of amino acids, that come out of the ribosome, and either by themselves or with the help of some other players in the game, they fold, and they always fold roughly into the same global overall shape. So the million-dollar question always has been, what is the secret recipe that allows a sequence of amino acids to always fold into the same fold, right? So your hemoglobin, my hemoglobin, that of a high-flying goose, or that of a deep sea diving fish are all very, very similar in their sequence. They all fold into the same fold, they all perform the same function. The tiny differences in amino acids in their sequence is what allows me to breathe, you to breathe, right now as we are doing, or the large amounts of time that a high-flying goose or a deep-sea diving fish will hold on to that oxygen compared to what we can do. Yeah? And that subtle chemistry is all that is required to give this vast difference in scales in living organisms. And it is exactly these kinds of simple rules, well, let's hope that they are simple, that lead to this vast spectrum of, this is just a snapshot of the vast spectrum of molecular architectures of proteins that perform diverse functions. Okay. So of course one can always, go back to this maverick genius, Feynman, who just said that you really could understand everything in terms of the jigglings and wigglings of atoms, which is what we will try and do now. Okay. So how do we understand this jiggling? What do I simulate? What is my workable model of an atom? Okay, and these questions become relevant. They are not very trivial questions, as we'll see in a second. Right? Because when I know that a set of atoms, I mean these are amino acids because we define them to be amino acids, but this is a set of atoms that fold into a fold. So if you put a molecule of hemoglobin, let's say one molecule into a test tube, the atoms do not go away, right? They stay together into a fold that's identifiable. So what is it that makes them stay together, right? And this is where you take recourse to the physical chemistry that you have essentially learned in high school and right? So you are modeling some aspect of reality based on certain empirical rules. And so this process began a very, very long time ago, right? When people were thinking of simple gases. So when people are thinking of simple noble gases, when they were trying to understand pressure, when they were trying to understand how the kinetic energy of atoms might relate to things like temperature, they are very simple models of just little spheres. Okay. But then the next step you can imagine, people started asking, oh no, but that's not necessarily true. When you start seeing crystal structures, you see atoms that come closer to what a hard sphere model will allow you to do. What's a hard sphere model? Hard sphere model is that you have one sphere, like in a billiard table, and another sphere. They will not go beyond their diameters. Whereas in structures, you suddenly find that there is deformation. So then people ask, okay, so in our models of this reality, what is the next bit in that equation we have to add to simulate this effect? And so on and so forth. And this process happened iteratively. Every time you think you had a good model of reality, along came an experimental observation that told you that no, this is not right. We have to modify it a little bit more, and a little bit more, and a little bit more. But the point over here is, how much do you modify, right? Do you need an accuracy to the 10th decimal place? Do you need an accuracy to the 100th decimal place? So the model has to be simple without trivializing it, but it cannot be too complex that you lose all computability, right? So what is that balance that you draw? And that's a question to which there is never a very satisfactory answer. Yeah? It's a very, very difficult question to answer, as I might touch upon later. But for our purposes, let's stick to these statements, right? First of all, I need to know to begin with how these atoms are arranged in space. So you may get it from your favorite technique, most notably is crystallography, X-ray. Now, of course, it's electron microscopy. You can have NMR. But now there's also enough data to build structures based on homology with known structures. What does that mean? That means the following. Let's say tomorrow you've gone out, found a strange organism which has hemoglobin in it. You've sequenced this hemoglobin, but you do not know what the structure is. But there's enough information known about the various sequences of the different organisms and the way they fold into the shape of hemoglobin that you can use this information, apply restraints, knowing that all the amino acids that are similar, the spatial relationships will be the same as you see in these other organisms. You use this information, and that is what is defined as building a structure based on homology, for those of you who do not know. If you know, apologies for having both. And then comes the key. The key is, how do I set up a model where the atoms talk to each other? What does that mean? Right? In simple terms, you can think of a positive charge and a negative charge. They come together. A positive charge and a positive charge repel each other as do negative charges, right? But they are talking to each other, right? So we need to think of a formalism to reproduce this observation. Right? And so that we'll call as an empirical energy function, because it is empirical. Yeah, there is no given over there. As I said, you build up this based on physical principles, very robust physical principles that you will not violate, but it's based on those, but nevertheless it's still empirical, as you will see. So where may these come from, right? So you may think of oxygen, trying to simulate an oxygen. In the simplest terms, you need an oxygen and an oxygen atom connected, right, by what is called as a covalent bond. So let's say we need this. Then somebody comes along and says, oh no, but now you've got water. So a model of water is one oxygen with two hydrogens, right? So there is, from spectroscopy, you know, there is some thing that is also called the angular bending vibrational, right? So you have to accommodate, and so on and so forth, and you keep adding these terms. And so these are the various terms that so far, in the kind of questions we ask classically, suffice for these models, okay? I do not have the time to go into details of these, but they're quite interesting as to why these things came about, how our older models of reality had necessarily to be replaced by these models, including things like van der Waals forces and all. And just to give an insight into it, it was this. This term, which is the Coulombic term, everybody thought, oh yeah, this is great, now we will use electrostatics and it will be the Coulombic term. The problem with this term was, when you have a positive and negative charge, then they should collapse into each other, right? Because the energy gets more and more stable as the distance decreases. And you had to stop this at some stage. And then people realized, oh no, they'll never come beyond a certain point, and that had to be defined by a van der Waals contact parameter, just to torture you at this time of the evening. But essentially, this equation, which essentially sums up various two-body potentials between atoms, meaning that you can model in two atoms talking to each other in your whole collection of atoms, just two atoms talking to each other, and that seems to be sufficient to at least help you understand and simulate the so-called covalent interactions that you see, which means things that are bonded. I mean, there are other things such as dihedrals and all, but they were brought into account for things like rings rotating, etc., etc. Okay. So once you have this system, then all you do is go back to your high school Newton's equations of motion and solve them. The reason that you can't just do it on the back of an envelope, where you can do it for an oxygen perhaps, but when you have something like a protein, you can't do that, right? Unless you are a genius. And therefore you require lots of very good computing, okay? But once you have solved that and you do all these various fancy sounding techniques, you get a very rich landscape in time and space of how these atoms as a collective would move and perform a function, okay? So something like this, yeah? It's a mess, right? You see, it's a mess. Okay, so what am I talking about? Well, what I'm talking about is at this point you use some very nice statistical techniques which seem to be improving every second week, and then you get something like this, okay? And this is your aim, to characterize the two things that you need to characterize about the chemistry of these molecules is the thermodynamics and the kinetics, okay? So the thermodynamics very simply tells you whether, let's say, you're familiar with a kinase. Let's say, I'll assume you're familiar with a kinase, okay? So the kinase structurally adopts an active conformation and an inactive conformation. Right? So the thermodynamics tells you what is the relative energetics of these two, right? And the energetics is based on that equation I showed you. And the kinetics will tell you how long will it be before one shape changes into the other shape. That is all you need to characterize to understand everything about the system, okay? And so this is what it allows you to do, right? So it allows you to understand both the thermodynamics and kinetics of things such as processes that are taking place at picoseconds, right? It is equally biologically important because you wouldn't be listening to me. Your neuronal activity very much depends on these kinds of motions, right? To allow things such as acetylcholine to be degraded by acetylcholinesterase on the one hand, and on the other hand you need very, very slow, large-scale motions which are allowing me to, for example, move my hand, these muscular motions, okay? How much of this can be captured in a simulation by itself? You may be familiar with the term molecular dynamics simulations. Those are the standard classical simulations. At the most, if you are very, very lucky and you have good computers, you may go up to a few hundreds of nanoseconds, maybe a microsecond. Beyond that, you need some sophisticated techniques that people have developed and indirectly try and surmise what motions at these time scales may encompass. Of course, the nice thing is because you have an atomic level description of your system, you can then tweak the system. Right? So you can perturb the system, observe the changes, and then go and hopefully have an experimental colleague who will test it for you and validate or invalidate, both of which are equally powerful. Right? Because whether their results come out to be positive as you expected, okay, you go and celebrate. If the results do not come out to be positive, that's a good thing because it allows you to first question the experimental technique itself. That is what you should do first as a good theoretician. But failing that, then you go back and then ask, okay, what is wrong in my model? Yeah? What can I do to improve my model? That's one. Or the other thing is, what am I doing wrong in interpreting the results of my model? Both of them are very equally very crucial aspects of trying to understand this. Okay. So, over the years people have been developing it, and of course it culminated in the award of this. I don't know how many of you are familiar with this, but nevertheless here it is. So, of course, they still refuse to say that it was for simulation technology, so they got away by trying to say that to understand and predict chemical processes. But that's because of a hang-up that, experimentalists traditionally had with simulation people. Okay. And so where did it lead people to, right? So, I think that the first major milestone was achieved in this paper that came out from a very good group. This guy is now the director of the Max Planck Institute somewhere, okay? So this was a major feat at that time, okay? And the major feat was accomplished because of a simple realization that because in these simulations, if you want to study a process, you need good statistics. What does having good statistics mean? Good statistics means that if you think that you want to study a particular motion, then you should see a few occurrences of that motion, right? You may need 10 or 12 different occurrences of that motion, okay? And that is what they managed to do at that time. They realized that we can do multiple nanosecond simulations, okay? At that time, believe me, it was a big deal. If you could do 10 picoseconds at a time, you had a paper. No, the first paper came out in Nature. Then many years later, they did 10 picoseconds, it came out in some big cheese journal. Then when people did 1 nanosecond, it came out in PNAS. Then when they did 10 nanoseconds, it came out in PNAS. Just to alert the public, I think, more like a PR stunt than anything meaningful at that time. But when they did this paper, you may ask, why was it published in Science, right? And the reason is because of this fundamental question they asked. That now that we can do a nanosecond simulations, what is it that we can meaningfully study, and what is it that can have a meaningful impact on biology of that particular process, okay? And so the system they chose was this thing called aquaporin, okay? So biologists, aquaporin is very important for maintaining homeostasis of cells. It selectively allows water molecules to go through, okay? So this simulation allowed them to do so because water molecules go past this membrane at about 10 to the power 9 water molecules per second on average, which is one event every nanosecond. So if your system setup is good enough, then you should see an event of a water molecule going past it, in your multiple simulations, and that is what they saw, okay? So of course the details are not that critical over here, but the interesting insight that they had was that it allowed them to understand why have these proteins evolved with a certain sequence, why is it that these proteins only allow water molecules to go through, and why is it that a small amino acid change over here can completely mess up you know. So for example, you might see people with limb swelling, retention of water, or cardiac problems. They are all related, at least a lot of them are related, to single amino acid changes along the length of this channel, and all it does is disrupt this smooth flow of water at a certain time scale. According to that organism, that time scale was important for its evolution, or whichever way it happened, right? Did that time scale emerge along with the rest of evolution? Whichever way you choose to see it. Okay. So that was the first major milestone, okay? Just to give you a perspective. The second major milestone appeared in 2004, okay? Now this by itself is a very, very trivial thing nowadays. But at that time it was very key in what happened. So what you see over here, if you can see that is, can you see? Can you see the back? So this is the close-up view of an enzyme, okay? And the enzyme is called HIV integrase, okay? At this point, HIV was like, you know, the biggest bad thing that ever happened on earth, and you know, everybody and their grandfathers and grandmothers were on the race to try and eradicate HIV. But the one thing in the life cycle of HIV that people could not perturb was the integrase, the thing that actually takes its genetic information and, you know, along the pathway of integration with the host organism. That they could not for some reason, okay? But companies had a lot of data on these molecules. They do something but they couldn't understand. That's in parallel. So Andrew McCammon, who is one of the big cheeses in our field, one of the ones who developed this field in San Diego, clearly one of his postdocs didn't get a date one night, okay? So this guy was just staring meaninglessly at a simulation he had carried out of HIV bound to this integrase bound to a molecule, okay? And then suddenly, you know, out of sheer boredom, something caught his eye, his or her eye, I forget. What had happened was, you see this molecule here, right, in green. That is where the original molecule sat. Occasionally he found that the molecule would flip over here, okay? Something you normally ignore. You think, you know, some kind of, not an error, but, you know, something that is not meaningful. Except that it happened enough times for them to stop and ask what's going on. And what they noticed was that during the course of the simulation, that being key, a little pocket would open up here, which did not exist in the structures, right? You may ask why didn't it exist in the structures, and that takes another lecture to discuss why it didn't exist in the structures, but it didn't exist in the structures. So if you take a protein structure from a protein data bank, you might see a cavity with a molecule bound to it, yeah? But believe me, there'll probably be other cavities that you do not see. And these cavities are a function of the temporal dynamics of the protein, okay? And a crystal structure is just an average snapshot, static snapshot. So they saw this, right? So they published this. This lady in, I think, Merck or Roche or something in Rome saw this manuscript, and suddenly, and to cut a long story short, they realized it suddenly explained why their molecules apparently seem to be working. And the reason was that these are long molecules that span both binding pockets. Because the company people were only looking at the crystal structure, all they had was access to this, so they thought, no, this molecule can never bind there. So something else must be going on. Suddenly this made sense, and suddenly, of course, it led to the development of these so-called butterfly inhibitors, because they are too flexible wings of these molecules. And within 3 years, FDA approved that drug. That's how fast that was. And that was remarkable, given that it just came out of a pure ideal speculation based on a pure MD simulation out of, you know, some bored postdocs life. So that, I think, was a very major milestone. And it began to give some confidence to people that, you know, there are questions you can ask that are sensible enough using this technology. See, that's the key. What is a sensible question to ask, which has both a spatial component to it and a temporal component to it? Which kind of time scales can I simulate? Which kind of spatial deformations or spatial changes, etc., can I simulate? That became a question. Okay. So now, of course, we have come to a stage where, you know, various labs use these kinds of technologies to ask questions such as the following. So you can ask questions at simple scales, you know, why doesn't my molecule bind? Why doesn't this peptide bind? Why doesn't these two proteins interact when I made a particular mutation? Or you may even go a step further and coarse grain your information, which means that Now instead of each atom in an amino acid, you represent an amino acid with one atom. Okay, and there are ways of doing this. And you can begin to ask questions such as how does a virus enter a cell? And I can tell you it does provide very, very meaningful insights into designing the next set of experiments. Okay. All right. Is that clear so far? Yes, no? Any questions? Okay. So now, so that is the technology we have. Okay. So where can we go with it? Okay, so there are many directions you can go with. Yeah, but we have limits on time. So I will tell you about a particular new form of probe that one can think about designing. Okay, and you'll understand what I mean by probe, but both as a reagent in your lab as well as a possible therapeutic. Okay, and we'll take the P53 as an example, only because P53 is a hot topic. Yeah, most mutations in P53 are not tangible yet in the clinic, but they are when a pathologist detects a P53 mutation, they feel very bad because most of those mutations are pretty damning. But there is some ray of hope, okay, and there is at least a ray of hope in some tumors in which P53 is not mutated but is wild type. Okay, so as a background to P53. When you look at a Caucasian colleague of yours who has come out from extreme sunshine exposure, you usually find that they have either gone red or that their skin is falling off a little bit, right? Is that right? Yeah, it's important. That is P53 in action protecting them, because those cells have undergone DNA damage. Those cells have undergone DNA damage beyond repair. So P53's role is to sense stress in the cell, UV radiation, get activated and ask the question, can the cell then be repaired? If it can be repaired, repair and go on. If it cannot be repaired, apoptosis. And then of course there's senescence and all, but this is in general P53's role. Okay. So P53 is that important, right? That's why it's called the guardian of the genome. But of course P53 being that important has to be kept at low levels. If P53 is always active, you won't be sitting there, I won't be talking. And so there are various enzymes that do it. One of them is MDM2, and in a lot of tumors this MDM2 protein is highly expressed. So P53 is unable to do its job. So you would like to ask the question, how does MDM2 control P53 levels, and can I do something about preventing MDM2 from controlling P53 levels in those tumors? That's the story. Okay. And so much has been done about this. That's the story. Now let's just step back and ask one question. Okay. So these three, for all the biology people here, at least cancer biology people here, know that the MYC-RAS P53 are three key pathways on which there is tons of very good information experimentally. Yet there is not a single drug in the clinic. In the clinic, there's lots in trials, but there's nothing there in the clinic available for patients where there are changes in these pathways. And the question is why? Why haven't we, after all these years, managed to do that? Okay? And the reason is the following. Let's just look at the two kinds of common therapies that we have. Okay? So we have small molecule therapy. Your aspirin is a good small molecule example, aspirin. Or you have large protein therapeutics. So the antibodies that I mentioned. So if you look at the small molecules, they are about 100 atoms or so, 500 molecules, or some 1500 molecular weight, let's say, at most. They are mostly oily, sort of, which allows them to slip inside cell membranes with ease. Remember, when you design a therapy, especially if your therapy is going to target something inside the cell, you have to worry about, is my molecule going to enter the cell or not? It's an unanswered question today. Right? It's a big challenge. How do these things get inside the cell? It's all right to assume that, oh, these are hydrophobic, they slip through cell membranes. Some of them do, yeah, but there are multiple other pathways that they take things inside the cell. Anyway, nevertheless, let's stick with this simple hypothesis for now. Whereas protein therapeutics are large, okay? They have mixed polarity of atoms, meaning they have polar atoms, non-polar atoms, hydrophobic atoms, and which is what prevents them from going inside the cells because they're that large. And therefore, if you notice, all antibodies, Herceptin for example, they all target extracellular membranes. Right? In the talk about the EGF pathway, all those antibodies are targeting these growth factors which are outside the cell. Okay. So, as an example, okay. So, quick back of the envelope calculation tells you that this only allows us to target 15 to 20% of the possible targets in the cell. It's quite depressing, really, if you think about it, that 70 to 80% of the targets are still beyond the reach of the two established classes of molecules that we have. Okay? And why is it? Because most things inside the cell are parts of relays, the cascades I talked to you about, okay, which are results of protein-protein interactions. And protein-protein interactions, by and large, are flat and large, and we'll see that in a second here. So this is a kinase, okay, in red. And here in yellow, you see the binding pocket with ATP sits. And so it's easy to make mimics of ATP, which will bind much tighter to this. And so one of the classic stories is Gleevec, which was thought to have been the magic bullet against a certain form of leukemia, until of course resistance arose, which was inevitable. So here you have a small molecule. A small molecule makes a few high affinity interactions. Okay? So what does it allow you to do? It allows you to play Lego with it. Here is a molecule, you sit with your chemist friend, then you design, okay, let's put a nitrogen over here, let's put a carbon over here, an oxygen over here, because I will tap into interaction that hasn't been tapped by that molecule, and that process works very well. All small molecule discoveries these days are optimized because of these kinds of modeling exercises, computer-aided design, for example, right? You can do that. Now look at this. This is Herceptin, okay, sitting on the surface of HER2, the receptor outside the cell. Look at this. Lots of weak interactions, but lots of them. Where do you even begin to make a change? You make a change over here, something else will go wrong here, and so on and so forth. So it's impossible to re-engineer an antibody from here over here based on the same principles that you use to re-engineer. There are other ways of re-engineering antibodies. It is quite successful, but certainly this is not so. Okay? And so you can see over here that while you have a hole over here to plug in your molecule and redesign it, over here there is a flat surface. There is no hole over there where you can put in a molecule and you go in and you can play Lego with it. That is the key to, and that is why protein-protein interactions inside the cell are very, very difficult to target with small molecules. Because small molecules go inside the cell, but they need a back pocket, they need a hole inside the cell. Okay, so what do we do about it? Okay. So here is a story of how you might try and think of new ways of tackling this problem. So let's look at this interaction. So this is, I told you MDM2 is the negative regulator of P53. Okay, so this is a crystal structure of MDM2. There is P53 or a part of it. Okay, and there is just conservation in this over 1 billion years. This is an antipathic helix. This is an oily pocket or sort of a pocket. One side of P53 has to be oily, so it goes and binds over there, but the other side has to be water loving, and that's how nature has been, right? So you have a peptide, it is water loving on one side, on the top there is water all around, and over here this oily surface meets this oily surface, job done. So of course you may ask, how would you make a small molecule discovery against this? Because you know that if you want to inhibit this fellow, you need to at least make something that mimics these key interactions. You know which are the key interactions because you have seen evolution, you have done experiments on mutagenesis, etc., etc., and you know that these are three key interactions. So whatever you make has to mimic these, and then you just need to do it much better. Okay? So people did the traditional way of screening, and of course they came out with this molecule called nuclein, okay, which bound over there, and if you compare the two, you see that the interactions are mimicked. Because it is a small molecule compared to a peptide, therefore it has certain advantages, entropic advantages that allow it to bind better. Of course, once people knew this, suddenly there was this gold rush, me too, me too, me too. And 10 companies got into this, me too will also design a mimic of this and make a small molecule, and they did, and there was great, much drum beating and share values going up until it all collapsed. Because it all badly went wrong because everything led to bad toxicity. Okay, which is inevitable for a small molecule. If you think about it, nature never had in mind that one day, someone will come and make a molecule like nuclein, right? So the chance that a molecule like nuclein of that shape will fit into a pocket in a protein, there's going to be thousands of such pockets, right? So it's not surprising that any small molecule that is designed, unless you're very lucky with specificity, the molecule will go and bind something else, which is also referred to as off-target effects, right? And therefore toxicity is inevitable. Okay, but nevertheless, this is the process of how people design. And now you see over here, if you look at these carefully, exactly that's what people did. Okay, let me add an oxygen here. Okay, let me add a C group here, and so on and so forth, to allow for things such as, you know, higher binding affinity, solubility, etc., etc. Okay. But we also know that in this story, if you were to cut out this bit of P53 and ask what happens if I just take this P53 as a peptide. Now this is part of a big protein, but this is just a peptide that has been crystallized. So clearly it must be binding. So let's ask the question that what does a peptide do? And so there are a lot of data. You can mature a peptide and go and get a very, very high affinity to 5 nanomolar. Okay? You can do that. So you know that you can do that. So how can it help? Okay? And what may go wrong in this process? So here is the essential question. The essential question is, here is a protein-protein interaction. Okay? The protein-protein interaction is mediated by this helix in this protein. This green helix is part of this protein. I would like to inhibit this protein in orange or gold or whatever it is. Right? So I'll ask the question that can I take out that peptide, that bit, because I know that's the only bit that interacts with this, and ask the question, will this act as a competitive inhibitor of this? That's the question, right? Like that. Will it? And the answer is no. Why? Because here the peptide is constrained in its shape by the rest of the protein. Now you have cut it out, so the peptide is flopping all over. Right? It's very disordered. But that has already given you a clue. Right? You may ask that, oh, but can I not do something to this peptide to pre-organize it in this shape? Can I do that? Yes, you can do that. Yeah? And there are many chemical ways of doing smart things. Okay? So one of those methods that has been developed over the last few years has been a technique called stapling, that some of you may be familiar with, and I'll tell you what it is in a second. Okay? So stapling is a technique where you do the following. You would like this helix to remain as a helix when it binds to this. Okay? So in this process, you want to constrain it as a helix, so you find two positions where you will replace the existing amino acid with this amino acid. Okay? These are unnatural amino acids. When you do this in your synthesis, then there is a ruthenium catalyst with one step. It clicks the two into place as a staple. That's why it's called a stapling technology. Okay? And now you have a helix that should act as a competitive inhibitor, and surely it does. Okay? And this has been demonstrated. So of course, where does simulation come in? Right? So just the history of the designs of these, I'll give you an idea that this was a paper that was published by the group who came out with that idea, a group by a chemist called Verdun in Harvard, and they did this. Okay. Now this is for the people who are doing computational work. Okay. This is great data. Why? You have the same length of the sequence, so you don't have to worry. You don't have that nightmare, oh, this sequence is one amino acid long, very short. There's some variation. You have a great spread of binding affinities. You have very, very good binding affinities, and you have horrible binding affinities, which is fantastic when you want to develop a model of how these things might work. Okay? So once you start modeling these things, you can very rapidly begin to get some ideas as to what are the determinants that make some staples work and some staples really bad and some staples are very, very good. And you know, just to cut a long story short, over here you see the staple is lying on the edge, and it derives energy by interacting with the surface of the protein. At this point you may ask, but your staple is all carbons, which is hydrophobic. But this is a protein surface. How does it interact with it? And that is when dynamics comes to your rescue. Because as this protein is fluctuating, right, these little pockets, these little tiny hydrophobic pockets just open a little bit. They see water all around, they close their eyes again. Okay? So you don't. When you see a crystal structure, that's why you'll never see them open. Because the crystallization medium is predominantly water. Right? But they are opening. And the moment they see a hydrophobic group in their vicinity, suddenly, you know, clump. They grab onto it. Okay? So this is nothing but a, I don't know, you call it some induced fit model, or you want to call it yeah, induced it probably is the best way to say, but anyway, you get the idea of what it is. And that is what drives this. But this is not a general principle. Initially we thought, yeah, this is very good, this is general. It's not. Just as whenever you think that there is a general principle, along will come something that, is an extreme value and messes everything up that you ever thought about. Yeah. But the good thing, of course, about these is that in hindsight, that, of course, you can just see that the similarity between the model structure and when they complex just 2 years later in a crystal structure, the similarity is quite close, and that's not surprising. There is no rocket science here, right? These days you could design something like this in an afternoon if you have your particular protein complex of interest, and if you have sufficient computing power, and more interestingly, the interest to do it, more importantly. But anyway, then of course you go on to the next step and ask the question that does it do things that it is meant to do? And you can see over here that of course it activates p53 in all the various assays, and for the more sort of experimentally inclined, you can clearly see over here that it does a pretty good job at activating p53 inside cells, and then it gets to its target in the nucleus, which is where you would like it to be compared to the various controls. And so, what does this allow you to do? So this now allows within reach a particular reagent. Why do I say that? Traditionally, what would you do if you wanted to investigate a protein-protein interaction in the cell and you did not want to kill the cell? Right? You can lyse the cell and then find out. If you didn't want to, you would go to sigma algebra, pick up some molecule that they say works, put it inside the cell. But you realize that while it inhibits your interaction, the likelihood that it will have a toxic off-target effect is quite high. So you are never sure that what you see is exactly what you intend to do. And what a peptide allows you to do is to take that lesson from nature about specificity and just add a little bit of help by constraining, putting in some constraints, and you suddenly have these things that one hopes that it will give you highly specific interaction. But the nice thing about this, so that's as a reagent, right? So now you would like to have something that you sprinkle on the cell, it goes inside and does. But the interesting thing about these are, if you think of it as a therapeutic, right, it's not just going inside the cell. The question you have to ask is, will it reach the cell at all? Why do I say that? Because the blood is full of proteases that are waiting to see these foreigners and chop them up, right? And so, this staple allows things to be protected against proteolysis also now, okay? That's a very, very interesting feature. And then of course, for some reason that is yet to be determined, these staples take the peptide inside the cell. It's a big field of inquiry as to what is the pathway through which these staples enter the cells, and it's an open challenge, so those of you who want to take it on. Okay. And so I know I just put this slide because this was one of the last pieces of work that was done. For those, some of you may recognize Sidney Brenner here. So he has a big lab there. So we worked with them to try and design a certain labeled staple peptide so that you can at least monitor it where it goes in the cell. He has this lab called Laboratory of Molecular Engineering. So anyway, you know more. So anyway, so yeah. So here it is. So now you have a great reagent. You certainly do not have the off-target effects and nothing. I have said less over here because it's a cell, right? It's a very complex environment. So I can never say never in this case, right? And yeah, anyway, so yeah. And the nice thing is it goes into physical. And so for those of you who are more biologically inclined, then you can go and ask questions about, will it work in tumor models of mice? And you can see over here that while it is not curing a tumor, at least, it keeps it stable in its volume and the body weight of the mouse doesn't go on, and so on and so forth and so on. And what was nice was this particular paper, that these people took that peptide from the literature, synthesized it, and they found that it could, with some kind of a transport mechanism, actually get into the brain. And that was quite interesting. They didn't have a mechanism, it just worked. But certainly, I'm sure that they are working on trying to optimize that. But seems to be a good thing. Okay. So, but there was one key point that I mentioned earlier on, right, which was about toxic effects. Right? But during that process, I made a statement of the following. I said that a small molecule makes makes a few very high affinity interactions. Okay, whereas a protein or a peptide makes lots of weak interactions. Okay, What does that tell us? That tells us that in a protein, okay, let's use this, okay. If you look at this picture, this is nuclein, okay. Nuclein makes a few high affinity interactions with MDM2. Now say I have removed one of these interactions, okay. I have removed this side chain of a methionine and mutated it to alanine. That one deleterious chain is sufficient to knock nuclein off. That is the basis of resistance, okay. So apart from things being thrown out of the cell, one of the key bases for resistance for all anti-cancer drugs is your very target mutates to rebind ATP, which is an ATP mimic, sufficiently, with sufficient efficiency that will knock out your drug but rebind ATP. Maybe not as efficiently as a wild type does, but sufficient for it to function as it does, okay. So the interesting thing about peptides is they are promiscuous. Apologies for using the word, but they are promiscuous in their interaction, okay. Because they make lots of weak interactions. So it doesn't care, right? If one interaction goes, then a side chain will just flip around and make another compensating interaction. And because peptides have this ability, and therefore it's not a surprise that a staple peptide works very, very well against a resistant strain of this. Then you can do experiments such as in vitro evolution, okay. So in a simple model, what you do is you create thousands and thousands of mutant strains of a particular protein, and then you test your molecules against it, and you find that staple peptides work. You may also continuously expose your cells to these molecules and then sequence that protein to see what's happening to it, okay. That was the big idea. As I said, that just when you think you've sorted everything, something comes along and messes the theory up. Unfortunately, in this case, P53 gets deleted, so it doesn't matter whether the peptide binds to MDM2 or not, and there's no solution to that, okay. But now, of course, there's a very variety. There's a lot of protein-protein interactions that have been probed by designing these by several labs in the world. Okay, it's still not hundreds of labs; it's in a few tens maybe, but it is a technology that's increasingly catching people's attention. The key is there is one company that came out of that work in Harvard, which is now in phase 2B clinical trials against certain tumors, and the results look good. But you know how it is, right? Most of these things fail in phase 3, but people are waiting to see. If phase 3 trials succeed in that case, then I think there will suddenly be a gold rush and a new Me Too era will happen. Okay. So where can we go from this, right? Since we talk of innovations, you may ask, okay, so you've done this, so what is the big deal, right? Sure, yeah? But then you may ask questions inspired by those observations. Okay, so hydrophobic cavities can open, they can grab things, okay. Now you go back to the drawing board and ask, how can I improve upon this without doing the standard design principles that I can do just by watching the structures? If you look at, let's say, your favorite protein which binds a ligand, some favorite protein P, and it binds a ligand PL. If you look at the crystal structures in the database, you may find the following. You may find that in complexes where the ligand is bound, in the unbound state there is no pocket over there, right? We thought about this earlier on, right? We discussed this. It's a hydrophobic pocket. If possible, it will be close to the environment because it does not like water molecules there. Yet you know that this ligand binds. What does this tell you? This tells you that the surface is malleable of that protein, and the pocket somehow must be opening. Either the pocket opens up transiently and the ligand is there and it just binds, either that, or else the ligand is just hanging around pushing things here and there, and somehow the door opens and the ligand binds. Nevertheless, the ligand binds. That's the key, right? Can I use this information to ask, can I induce pockets to open up? Okay? How will you do that? So this is how you will do it. So normally for those of you who carry out computer simulations, what do? You will take your protein crystal structure, you will surround it with water molecules and add whatever ions, etc. For those of you who do it. That's the standard method of doing it. And then you carry out the simulation in a manner I mentioned, and then you watch what happens. Now you ask that if there are hydrophobic cavities, how do I induce them to open? So you ask a completely meaningless, totally unfeasible question. At least a chemist will probably, get very upset with these suggestions. You infuse your system box with a few hydrophobic molecules. Let's start with benzene and ask what happens, right? This is what happens. you will suddenly see that little pockets open up and benzene will go and sit inside. Okay, and this you can do with your favorite little molecules. And there are a few groups now who have developed these techniques for the various kinds of fragments as well. Okay, so what can we do about this? So we can ask the following question, that we had this particular staple peptide design. Can I induce a pocket in its vicinity somewhere to add another amino acid to get higher specificity, higher affinity? Is that possible? Yeah? Let's try. Yeah? There's no harm in trying it. And so, of course, just to cut a long story short, then this PhD student developed this algorithm to do this, and sure enough, in the vicinity of that, there was a pocket which you see over here. And so, what do? It was a benzene. So, the simple-minded addition you will do is a phenylalanine ring over there, right? That's the simplest, mindless thing to do. And so, that's what we added, and then we simulated it, and sure enough, and then, so this was predicted, and this was the crystal structure that was finally resolved by some, people to whom we are very grateful. And you can clearly see that the match between that prediction, that was very interesting, that suddenly gives you another tool to further optimize the kind of ligand or peptide or antibody design that you wish to do in a manner that traditionally we were not accessing. It was always there, right? It was just a matter of developing a methodology to access this tool. Of course, having better computers helps this process because you need to do lots of these simulations for statistics, of course, yeah? So, anyway. All right. And so, so this peptide work led to, sorry, I've just circled two people. For those of you who recognize Dilraj Lama, he was the person who was here, who has done a lot of work on this, this thing called translation initiation factor, EI4. It was in one of your slides earlier on. And then, of course, we have a lot of work on designing these new peptides. Okay. So then somebody will ask you the question, okay, you have a staple peptide, but the edges are still exposed to proteases. What do you do with that? Right? The edges will still be exposed to proteases, so it will still not be as stable a molecule as you would like it to be. So then this guy, who you can barely see, with him, he asked this question, okay, given this topology of this particular binding pocket, can I make every amino acid into a D amino acid? If you can succeed in making every amino acid into a D amino acid, your job is solved, right? Because proteases will not chop up D amino acids, okay? And so therefore, just to cut a long story short, this ended up in, then you can play around with different kinds of staples, so you can make all D amino acids, you can add various kinds of staples, and then after that, it's just a design principle. A very collaborative chemist who will happily synthesize it for you, or you have lots of money to get a company to synthesize, and then various people to try and test it for you. But then you come to a stage now where if you're seriously thinking about developing these as a therapeutic, then you should set in place a series of collaborations where you can ask very, very important questions such as this, okay? So, okay, CD spectroscopy is easily available, right, these days. You can find out, or NMR, you can find out roughly what the conformation is of your peptide, okay? Interactions you can do using fluorescence polarization, ITC, SPR, whatever, that's also easily available. What starts becoming interesting is that your cellular activation, once again, no big deal these days, for biologists, that you have to start asking the following two questions which are very critical. One is LDH release, okay, which essentially is this question that was asked when our first report of this work was completely thrown out by the referee. He asked a very simple question. Rightly so, he asked, how do you know that your molecule is not perturbing the membrane long enough to result in activation of a stress response pathway which gives you the activation of P53, because P53 is the stress response protein. I never thought of that question, and absolutely he or she was very, very right. It was unfortunate that they rejected the paper, but nevertheless. Then another year passed by, and then you have to design these things, which is not anymore, I think it's quite easy to design a setup whereby you can ask that, is your membrane being perturbed for long enough for systems to leach out, or does the system just go past? So you have to do the LDH release assays, and then you have to do what are called counter screen assays. So counter screen essentially is off-target effects. You want to make sure that your activation is because of P53 being activated as a result of MDM2 inhibition and nothing else. And a simple way is to look at either a mutant system or else a system when there is null for one of those two proteins, and then you can figure it out. But nevertheless, it becomes very, very important. So, and then once you have done that with a series of your designs, then of course, hopefully you'll get lucky and you'll come out with this. Okay. So that is that. So do I have 5 minutes? How much time do I have? 5 minutes? Okay. So I will just tell you, since there's a lot of excitement about people developing companies, just to give you an idea of the importance of multidisciplinarity in this day and age. So a lot of it was reflected in the earlier talk. I'll tell you a quick story about what happened, since it refers to the VEGF pathway. Okay. So this just took 8 months. We had very little money. So we had to be very, very careful in how many compounds we got synthesized. Now the traditional method would be this. Okay, so if you forget all this, okay, this is important. Okay, so this tells you that this angiogenesis that leads to the leakage, which you gave a very nice explanation of earlier, at least for those of you who attended. For those of you who didn't attend, then these pathways get activated, VEGFR, PDGFR, alpha, beta. Okay, in some angiogenesis processes, which leads to leakage, which allows for deformation of your vision and eventual blindness. So if you do not treat it, then you essentially are messed up. And this is the treatment. Okay. And just to mess up your life some more, I have a fantastic video which makes me cringe every time I watch it. But I will just go very, very quickly to the really horrible stuff. Okay. So this is the only treatment available. Watch. Are you ready? So my sister-in-law gets it, so it's not painful, she says. There, you see? That's an injection. Do you see the injection? Shall I say it again to wake you up? Just in case you have... See, stabbing, literally stabbing. Anyway, pretty horrible, isn't it? Anyway, so stop. Right. Anyway, so I think that will just be easier to. So that is what it is. Okay. So this only works for about 30% of the patients. So my friend's mother is one of those unfortunate ones where it doesn't work. Okay. There are these 3 antibodies. They work on these growth factors. You see how many growth factors activate these receptors. And these antibodies are very specific. So it's quite possible that part of the reason why they are not so effective is because they only target one or few growth factors. And then he showed in one of his slides that once you start targeting the receptors, you seem to get a bit more effect, right? So the project really started off not with this, it started off with trying to target an unmet disease, a particular form of cancer called CMML, whose target is PDGFR. Okay. So this guy developed a very, very ingenious technique, which he won't tell me how it works. But nevertheless, what he does is, he calls it humanized yeast. Okay. He engineers yeast. He takes your target and puts it in the yeast. And in contrast to traditional models of screening, where you throw in molecules when they inhibit your protein, the yeast dies, here the moment you turn your particular target on, the yeast dies. Okay. It's only when you inhibit the molecule, specifically your target, that the yeast lives. Okay. That is his patented technology of humanizing yeast. So we did this with a bunch of about, I think we had only 500,000 library of molecules, which is quite small compared to what a company would normally do. Millions you would screen. Okay. And so this is the model, right? So you find some hits, and then you very quickly try and figure out what it is how it is that this small molecule is targeting that particular protein. And in the process, by homology, you discover that it also should target the VEGFR pathway, even though it was for PDGFR. And sure enough, when you do the test, you find that, yeah, it does target all of those 3. But anyway, to cut a long story short, we synthesized only 6 molecules. That is all the money we had. We were lucky, I must admit. Either we were lucky, or else this humanized yeast is an absolutely amazing system. We do not know the answer to either of these. We prefer to say we are lucky. But essentially this is what has happened in the end. So these molecules are now sub-nanomolar within about, as I said, within a year and a half, based on this iterative process of design and testing. And you can see that compared to in vitro screens, you can clearly see that there's a lot of no's in these, physical binding, avoid selection of toxicity, cell-penetrating hits. Whereas over here you got a yes in all of them. I mean, essentially it satisfies a lot of the conditions you would require for drug discovery. Okay, so yeah, then you model it in, and then you remodel it to improve the affinity. So, and now you can see that we have some sub-nanomolar inhibitors, like almost sub-nanomolar inhibitors of the same. Now, here is an interesting thing that really puzzles us about the ability of yeast to do this. Okay, so you mentioned sunitinib, I think, in your talk, right? And then there is another method. What this is the number of off-target kinases that these drugs which are in the clinic hit, by the way. Just to tell you how dirty these drugs are. If you were to think of them as dirty, there are other reasons why they work. They are therapeutic windows and all, but if you were to just do what is called a kinome assay, where this company for a certain amount of money will take a molecule and test its affinity against 500 or 300 or so kinases of the human kinome, it will give you these circles represent the affinities, right? So you can clearly see this is very, very specific. There are a few, of course, If it was just over this, I would be very worried that, is that clean? And here is some mouse data. So they allow the mice, they poor mice. They mess up the mice's eyes with lasers, and then they allow it to regrow back. Right? The mouse regains vision, but of course they kill it, unfortunately. And so you can clearly see over here that the drug works. But here's the interesting thing. So then we made an eye drop based on a certain formulation. And it is very clear that the eye drop works. And that is the basis of this company right now. Okay? So it's quite pleasing so far, right? The mouse data is quite good. But of course, if people had mice as pets, it would be a very successful company. But as such, now people don't keep mice as pets. So we don't know whether it will work in human trials, but nevertheless, the process is on. Okay. And so finally, there is the last slide, since precision medicine was mentioned, and what is this relationship to our, the kind of computational modeling you can do. So you can ask this question based on this population sequencing. Okay, the population sequencing is becoming very important. Okay, and so there's a very nice effort locally that the government funded in Singapore, which was to ask this question that all the databases that are used for reference in the world are based on Caucasian information. Yeah, they're heavily biased towards the Caucasian genome. Whereas, countries like India, Singapore and all should not be biased like that. So what can we do about it? So Singapore being a small country, it was easy to set up a large-scale sequencing effort, which was just completed with the 10,000 local population made up of Malay, Indians and Chinese. Okay, and all the information that is coming in is now being put together in terms of the variance research, so single nucleotide polymorphisms, at least at the first level of inquiry, into asking questions that if you have Gleevec now for a patient from Singapore or India or Malaysia, where are the SNPs lying with respect to where this molecule is supposed to lie, and how might it perturb the efficacy of this particular drug? That is one of the powerful aspects of this sequencing of populations that is going to materialize into a therapeutic index of some sort, help clinicians, but it has to require a very, very large scale multidisciplinary effort. Okay? Okay. So I think that is it. This is the kind of effort, I think, which you can set up. I think you have all the ingredients. Maybe you already have set up, I don't know. This is how we do it, at least in our institute, where we look at all these things, and then we have experimental collaborators in all these labs in IISER, in the various institutions and clinics in Singapore, and then funded thankfully by Verma. And then of course, there are a lot of interactions in the world that we have. I'm just mentioning some Indian ones over here. The interesting thing about this is to have people who are convinced that there is power in this kind of modeling. That is what you need among your experimental collaborators. And you need very motivated clinicians now to take part in this, but it works. It's a workable model. Here is one example. I'm sure there are hundreds of examples in the world. This is not unique. And so here I will finally thank the various people. I must thank this person. He taught me a lot about P53. For those of you who don't know, he was one of the 3 co-discoverers of P53. And this acts as a great bond between us, catalysis. Singapore, and I must thank the various funding agencies. So more recently, I've put this in blue. So what this stands for is, so we had a honeymoon period, right? We had a honeymoon period for 10 years of unlimited core funding. Okay, you know core funding? Sorry. Okay. No, I didn't know core funding till then, right? And suddenly we have all this money to do. And then suddenly, the honeymoon period ended. And now we, of course, join the other mortals in trying to, fight against grants and the rejections of grants, rejections of papers, every second day. So the government put together the following. They said, you go and your industry to help you. And if they are interested, if they think your technology and your approach is good enough, then we will fund you. So there are these industry alignment funds pre-positioning. Okay? So when you have industry interest in your project, like we had luckily enough from Merck, then you get a lot of grants. And so these grants allow us to take part in a lot of programs. And I think it is a workable model if you can find industry who is willing to, pump in that kind of a money. So, yeah? Okay, thank you.
Thank you for a very exciting talk. I have a couple of questions, but I'll restrict myself to two. First, regarding your yeast model, were you talking about yeast-2 hybrid in terms of interaction, protein-protein interaction, and if it is? No. And then do you think that within yeast, if the interaction is happening, it also recapitulates in human cells?
So everything is repeated then. All the work, I didn't have time, so everything is repeated. Once it's shown efficacy in the yeast, then it's completely taken to all the various human cell lines that you need to test it to. So now all this work is done by CROs, we don't do anything. Okay.
And my second question is more on simulation of microenvironment. So you talk about simulation of cell and then proteome or maybe genome, but can we also, or have you tried simulating microenvironment which is also impacting, let's say, AMD or tumor progression?
Only in terms of the identifiable protein components, not at the level of the next scale, where you're doing a more coarse graining of the actual environment and the sort of effects that you mentioned, right? The effects of mechanical stress, the effects of multiple proteins having an effect, that is beyond the scope of these kinds of computational modeling exercises. You can do it in what is defined as a systems biology context, right? You can write many equations and things like that. But in this atomistic level representation, we are essentially limited by the complexity of the system, by the resources that we can access. I mean, I think more recently, in a couple of weeks ago, was the first large-scale simulation done after years of either the light harvesting complex or one of these large complexes, but just by itself. You can do coarse graining. So we do the whole dengue virus now as a whole virus simulation. But still it's just a virus. With a coarse grain virus with a coarse grain model, let's say, of the surface of a membrane to try and figure out what may be the key steps in a particular interaction that lead to an observation. But the technology is still not there to answer the kind of question that you raised. It will happen sooner or later. Maybe quantum computing will happen. But right now,
I have a short comment. So I haven't shown that data, but we do have a data where we have shown it. We put retinal pigmented epithelial cells, which are key in AMD, on like stiff and soft surface, the secretion of cytokine which leads to wet AMD changes. And the response to Avastin, I think that was the Avastin injection which you were showing.
So one of the things I didn't say is that these molecules are targeting the intracellular kinase domain, by the way. That's another key difference between the two. But it might have the same effect. So thank you.
Another question over there. So my question is regarding the structural prediction of disordered protein. So how reliable is the predicted structure, in silico predicted structure of this disordered protein? And usually these disordered regions play a crucial part in the binding pocket of, and when you target your drug to those binding pockets, so there might be more non-target effect, or the clinical efficiency of when it comes to the nucleotides will hamper that.
Right, yes. That's a very important point. So the first thing over here is that you asked a question, the prediction of the structure of a disordered protein, right? So now we move away from a structure to structures. You have to think in terms of ensemble. And when you think in terms of an ensemble, then you start thinking in terms of statistically which is the more prevalent set of conformations. That's one. Okay. The specific answer to your question about this particular system is that you do realize that it is biased by the information we already have. So the drive for those 3 amino acids to target that pocket is so high that any amount of disorder in the rest of the regions do not seem to affect it. I say this because if you do an NMR structure of this, you always find that that region is always helical in the NMR. Okay, that's important. Now when it comes to how does that relate to specificity, you are absolutely right that it might go and bind. And that is why you add these new technologies. Can I open a new pocket somewhere else to add another specificity handle onto it? Yeah? That's the idea, that you use these technologies to try and improve upon specificity. Because yes, the cell is a very crowded environment, the cell is a very diverse environment. And the chances that anything will go and bind to 3 other things is quite high anyway, right? And there's nothing I can do about it. Except that within your target you start keep increasing the specificity. Just like it happens in antibodies. If you think about it, why are antibodies so specific? It's because they're just making these, either these large extended surface interactions or else sufficient islands of interactions that allow them to get this specificity. So I think there is going to be some confluence from antibody area into this peptide design anyway. Right? So actually, I don't have time, but we have this thing on cancer imaging, and it's exactly what we are doing. We are taking what natural antibodies are telling you about and trying to use that information to design various kinds of peptides which can then be tagged on to one of these, probes where you shine a light and suddenly light up and say, yeah, this person is going to respond. And the reason for that is the following, that when I mentioned that just like wet AMD, you know of immunotherapy, right? It's a big thing now. So in immunotherapy, again, it only works for about 30% of the patients, right? But how do you find that out? You inject the patient with a $100,000 injection, and only then you find out, oh dear, I'm sorry, it doesn't work, right? So the idea is that if you can find whether the target that you are targeting with the immunotherapy is expressed in a patient by a probe, like one of those things you drink before you go for one of these scans. So that's the idea here. And the design of those peptides with those probes is being motivated by what natural antibodies are already telling us with the specificity. So, yeah.
So we have time for one short question. If anybody? Okay.
Like how much detail is necessary for simulation? Like details of expressions in modeling.
Like, sorry, could you repeat that?
In modeling, we have physical expressions of interactions. How much is necessary to predict the?
Right, good question. So it comes back to that point in the beginning that we would like to keep our model simple, but it is determined by the question I asked. Okay, so if you just look at this picture, this one. So there are 2 things here. One is ATP, which I know is involved in catalysis, and one is glider, which binds, right? In chemistry terms, there is no reaction taking place here, although there is a reaction anyway when 2 things bind, it's a reaction. There is over here, catalysis takes place. So if I just want to understand the physical binding within course of this and this, that equation I showed you is sufficient, give or take a few things. If I want to understand how the phosphate gets transferred to a substrate protein, this model fails. Okay, this model will fail because of that very first equation that I showed you. So we didn't have time to go into that slide takes 4 hours to go into detail. But it's quite interesting how the development of it took place, but I'll just take a second. You see this, right? This slide. This one. Yeah? So these models You see this is a harmonic model. So if you imagine using this model as a bond model for phosphate transfer, it will never work. Why? Because however much you extend your bond, the potential energy only keeps increasing infinitely. It's never going to break. So you model it by using some different things called, there are some Morse potential. It's flatten off at a certain distance where you know the reaction takes place. So that model doesn't work here. So it comes back to the point that what is the question you are asking? That is the key. But for classical things, if you want to just understand how conformational changes and all are taking place, this by and large is surprising how well it works. When you start going into details, what is the effect of a potassium ion or a sodium ion, then your nightmare begins. So.
All right, so let's thank Professor Verma, and before we end, I'll request our director to hand over a memento.
I'll leave it in the hand.
Thank you. Thank you very much. So those of you who are attending ABN now can proceed to